Hello everyone, my name is Colleen Lezovitz and I'm a Senior Marketing Manager at Worldwide Clinical Trials. I would like to welcome you to today's webinar, Accelerating Analgesic Development by Optimizing Early Human Models of Pain. Before we kick things off, I did just want to go over a few things. All attendees are on mute, but please use the Q&A window to submit questions at any time pertaining to the presentation because we will be reserving time at the end for a Q&A session. This webinar is being recorded, and tomorrow all attendees will receive an email with a link to access the on-demand recording. Our speakers today are Dr. Miroslav Bachakonya, Worldwide Senior Medical Director of Neuroscience, Medical and Scientific Affairs, and Dr. George Asti, who is the Vice President and Medical Director of Worldwide's Early Phase Services. At this time, I would like to hand things over to Dr. Bachakonya. Doctor?
Thank you. Welcome everybody to this presentation on the topic of advancing analgesic development, and in this case we'll be talking about the role of human experimental models as one of the ways to make that advancement. Our agenda for my part of the presentation will cover recent history of analgesic drug development and challenges. We'll discuss what is in common language considered experimental, the clinical models in drug development, primarily disease models during the phase 2 and 3, which is in contrast to experimental models, which will be focus of today's discussion. And we'll see how we can march to new paths to development of new therapies. We'll also review reasons for recent failures, and then we'll proceed with the discussion on human experimental models of pain as the solution to these challenges. And up to my and Dr. Ati's presentation, we'll have a Q&A. So what's the status of drug development in the recent past? On the one hand, there is a pretty large unmet need, and that's probably best summarized in the report of Institute of Medicine that was published in summer of 2011, titled "Relieving Pain in America: Blueprint for Transforming Prevention Care, Education, and Research." And it's right in the front of this large report of 50-some pages, epidemiological statement that over 100 million of Americans suffer from pain. To the most part, majority of these patients do not relieve appropriate relief from current therapies. And another sad fact is that in the past 2 decades, an area of interest of mine, such as neuropathic pain, there was over 150 studies and clinical trials, which resulted only in approval of 5 new therapies. And actually, if you really scratch the surface and look, there was only one therapy that was truly novel, since the other therapies were repurposed from other therapeutic areas. The only one that was completely novel was the conatide, which is peptide. So, considering this status, it's unsustainable to move forward in drug development with such a high rate of failure. And even therapies that are approved, if one looked at what are the characteristics of the trials on the basis of which the drugs are approved, turns out that on average only 40% of patients receive 30% pain relief. In reality, that translates that patients' pain decreases from 8 down to 6. So in reality, 6 on a 0-to-10 scale is still a lot of pain that would be considered moderate to severe. So even patients who get the benefit from these therapies still suffer from a great degree of pain. So in reality, I mean, this is probably one of the simplest and best examples how inadequate our current therapies are for what appears, it's clear, a large number of patients just in a single country such as the United States. And this epidemiology has been investigated in other countries and pretty much parallel with the rest of the world. So what are the models that are used during the analgesic development? Most commonly, probably most of the audience is probably familiar with the acute pain models such as molar tooth extraction and is the most common one. One model that they can also often and been recognized is bunionectomy, as well as perioperative pain. Probably newest kid on the block is liposuction. If we look at what are the characteristics of these models, and in general, models are characterized by a great degree of variability. And dental model is very short in duration. Usually pain is mild to moderate, so there's very little room for testing the efficacy, because for the pain that's mild to moderate, it's very easy to see the placebo rates to go high. Bunionectomy pain is a little more intense, so one might have a more assay sensitivity. And one of the characteristics of perioperative pain, it has a very high degree of variability, and frequently patients receive concomitant medications, which is in many cases difficult to control. And liposuction is the newest model, and probably is more stable, and pain is a little more intense. So with all of these characteristics, there are opportunities to use these models, but one, again, has to be cognizant of the limitations, and these are some good examples of what they present. And one of the first challenges is which model to choose, and there are certainly challenges when models are chosen for phase 2 and phase 3 trials. Choice of human clinical disease models is usually based, to the best of my observation, on a guesswork. And most often what happens is when new studies are being considered, we always go back to what was traditionally used. Those ranges based on preclinical rodent, most frequently rodent models of PK, without most often and sometimes with PD readouts in some of the rodent pain models. But most of this is really decided when moving forward in phase 2 and phase 3 on very sparse data. And then when deciding which outcome measures to use, again, it always is borrowed from previous studies. So challenges are pretty clear, and the next question is how to deal with them. In regarding the models that are used for phase 2 and phase 3, for chronic pain models, there are a few, although they all present different opportunities, but frequently there are more challenges than opportunities. What has been by far most studied is postherpetic neuralgia and diabetic painful neuropathy. And although it's considered, for example, that diabetic painful neuropathy is one disease, etiologically it is. But if you talk to any of the clinicians who deal with those patients, it's pretty clear in discussion that there's probably more than one phenotype, more than one type of pain that these patients experience. And that might be a good explanation why so many studies utilizing the diabetic painful neuropathy model have failed. So again, just one example how something that looks on surface straightforward and simple, and again, frequently used by sponsors when submitting the proposals to the regulatory agencies for drug development, have been really a steep hill to climb. And probably similar is with post-hereditary neuralgia. The other thing when doing the drug development with these disease models, they have by now approved the therapies, and in case of post-hereditary neuralgia, there's the approved vaccine that has really had a significant impact in terms of decreasing number of people experiencing shingles and consequently post-hereditary neuralgia. And in such an environment, it is getting increasingly difficult to study these disease models because those patients are fewer in numbers and also frequently they are on concomitant already approved therapies. So for them, there's very little incentive to go on into blinded placebo-controlled trials. So certainly a significant challenge. And another disease model is osteoarthritis, which is probably, from the model development, simpler and more straightforward. But then the question is, osteoarthritis or what? And frequently there are probably less relevant for mechanisms or either the disease or of the drug or intervention mechanisms, rather than that being the primary reason to selection. Frequently, selection is made on opportunity to recruit patients. So again, there is a mismatch as a challenge. Migraine headaches has had specific development path, and probably this is in pain disorders, brighter entity that has been successful in developing therapies not only for acute treatment of migraine but for prevention. And maybe should be used as an example how to match mechanisms of disease with mechanisms of the action of the agents that are being studied. Again, should be used as a positive example rather than, as I discussed, diabetic neuropathy and post-hereditary neuralgia where things frequently fail. Low back pain has been challenged primarily for reason of being more than one disease under one umbrella. Fibromyalgia is an interesting story in terms of being a model that was something that most of the investigators have been hesitant to embrace as a model, but with a series of successes in terms of approval of the therapies really has helped in us understanding the disease itself. So a number of different models presenting and illustrating to us opportunities and challenges. But again, in the case of fibromyalgia, one of the issues is the outcome measures. And again, for those who are interested in this topic, you will find that certainly there is a big discussion what to look there. Post-traumatic neuralgia is a topic of recent interest, same as small fiber neuropathy. So, but again, this is when the decision is made to go into the clinic. But the question is, how do we make a decision which of these models to choose? And again, this is just a brief summary of what this audience knows very well. We started preclinical models as a scientific foundation, and this is done on cells and cell cultures and rodent models and other animal models. And then we go to humans. And in phase 1, we use pharmacokinetics, PK, safety studies, and usually not much is done at that point. and then we go to proof of concept studies in phase 2, and then pivotal studies in phase 3. But going from preclinical to clinical, from rodent to laboratory studies to human studies, really comes to crossing a big chasm. It's a leap of faith. And I think rather than jumping over the cliff, over the abyss, we should build some either bridge or trellis or some way to cross this in a more sensible way rather than most likely ending in abyss. So if you look at what are the probable reasons for failures of trials, in the beginning identified failures of trials in phase 2 and phase 3. And it's most often uninformed choices of clinical pain disease models, which is usually chosen on the basis of traditional and so-called established paths. Those are based on preclinical animal modeling and not on human PD information. Therapeutic range is not clear until we get to the phase 2, and again, frequently not explored fully. And additional reason probably is that outcome measures used in trials are not based on any human data, but rather on established, so-called established models, in spite of the fact that new chemical entities frequently have a new mechanisms of action and probably require new ways of exploration and further development. So this is where human experimental models of pain come in. Well, question is, what's the foundation? What's the background and rationale that human experimental models could be used? One of the things that is the opportunity for employing the human experimental models is that outstanding mechanism of acute and chronic pain are peripheral and central sensitization. And the other thing is that outstanding symptoms of acute and chronic pain are manifesting in the form of allodynia and hyperalgesia, which is also something that human experimental models can utilize and assess a range of these symptoms in signs with manifestations of peripheral and central sensitization. So this is taking opportunity of what we have learned from human research. This is next slide. A pretty kind of quick and simple sketch of what this is referring to. In the case of any incident that will activate the nociceptive system on the left-hand side of your screen, the simple events such as touching a hot stove will activate the peripheral and central nervous system. And although we think in terms of pain as one single event, actually it's a series of physiological phenomena. And without getting into great detail what those physiological phenomena are, as I said, in most terms, we are considering these as manifestations of a peripheral sensitization and a central sensitization. For those who have been studying neuroscience would remember that indeed there is a division between peripheral and central nervous system, and there's a different series of physiological phenomena that in case of the nociceptive system have been very well defined and clearly distinct. So why would we do human experimental models? And when would we do that? Well, best opportunity is to do this during the phase 1 when it's critical to obtain information that can help in decision making and for planning phase 2 trials. One of the things that human experimental models can do, is actually the only approach, is to add PD for a new chemical entity during phase 1. We can also answer the question of whether NC has any analgesic properties in humans by studying the effect of the new chemical entity on allodynia hyperalgesia that we induce using human experimental models. We can also look at the dose-response effects, and we can also determine whether there's a predominance of peripheral central effects of the new chemical entity. So what are the human experimental models? It's not one. It's actually one could consider a battery of tests that could be employed. And one of the main characteristics of human experimental model is to do sensitization, because referring back to 2 slides previously, what we do is actually sensitize for the limited period of time on the nociceptive system, and then we apply quantitative measures and quantitative sensory methods to study that sensitization. And on the basis of the quantitative assessment, we can see the effect of new chemical entity on that system. The sensitization could be induced with compounds such as capsaicin, menthol, mustard oil, and we can also use physical methods such as UVB burn and electrical intradermal stimulation. We can also look at the actually workings of the central nervous system using cold pressor test or a controlled pain modulation. And so there is a variety of methods that could be applied. The latest development has been laser selective stimulation, where we can do the psychophysical aspects of this testing as well as laser evoked potentials, where we can selectively activate and study A delta and C fibers. So what are the characteristics that we can, in the measures we can capture during this period of sensitization? Certainly we can look at ongoing spontaneous pain if it is a part of the disease, not the disease, experimental model, and then we can apply a variety of types of stimulation in the form of conscious sensory testing, and we can apply either mechanical or thermal stimuli. So one of the experimental models is UVB burn, where we can induce under controlled conditions and circumstances transient burn, usually in a 1 square centimeter area, what by all purposes would be a 2nd-degree burn, which resolves within a couple of days, so it's transient. But during those 24 to 36 hours, we can do the testing, applying the quantisensory testing, and look at the different forms of allodynia hyperalgesia as the manifestation of this. And one thing that's neat about this model, it is predominantly driven by peripheral sensitization. So this is where we come by applying this model. We can test the agents that have a predominantly peripheral effect. One of the models is intradermal capsaicin, where capsaicin is injected intradermally, as demonstrated in this a picture on the right side of your screen, where there's a site of injection, which kind of leaves like a little blanched site, and then there's within a matter of minutes what's called axonal reflex flare, and when tested this area, then we can identify area of secondary hyperalgesia, phenomena that's a manifestation of central mechanisms. And dermal capsaicin is performed by applying capsaicin at the dose of 100 to 250 micrograms, and then we can measure either pain on injection or by applying different methods of stimulation, look at either mechanical thermal hyperalgesia and allodynia. This model is shorter in duration, it's 2 to 4 hours, and this provides the time period where we can look at the effect of a new chemical entity during this time period. Cold pressure test is also frequently used, but as opposed to looking at the peripheral mechanism, in this case, we would be looking at the predominantly descending modulation and inhibition. The outcomes that we measure using human experimental model of cold pressure is pain intensity and pain tolerance. So which of these models to use really comes down to the answering the question, what's the question? And the question is most frequently, can my analgesic intervention, being drug or device, can I demonstrate the efficacy of this intervention? And probably the best way to look at it is to ask the question then, what do you know about your new chemical entity or device mechanism of action? So that way you can match the mechanism of action with the mechanisms of the disease or model that's being considered to be affected. And so one way to approach this in a systematic way is to apply not one, but probably 2 or 3 models to investigate fully and comprehensively in terms of determining whether the new chemical entity or intervention would have effect. So in this table, we have a UVB, intradermal capsaicin, and cold pressure test as a method of sensitization and study. And then if you look at the drug classes like NSAIDs and opioids, they've been very well established that, for example, for NSAIDs, they have effect on UVB, but not intradermal capsaicin, and cold pressure test. On the other hand, because they have a peripheral and central nervous system action. And then gabapentin has a predominantly central action without affecting the UVB much. So then if we have a new chemical entity, when we would apply it to these models, depending on what results we get, we will then know whether the new chemical entity has a peripheral or central effect. And that way we would answer the question, what is the mechanism of action of this new chemical entity? So how do human experimental models compare, and how do they fare when compared to clinical pain models? There has not been a study to march through the whole process, but this review in the British Journal of Pharmacology is a good example, and actually pretty comprehensive review, how clinical models and experimental models are highly correlated. And one thing that's encouraging is that not only the positive results from experimental models go in the same direction as clinical models, but also in cases of negative outcomes, they go in the same direction. So conceptually, there is a support for this approach. So to kind of conclude and summarize, what is the benefit of using human experimental models of pain in phase 1 and at the time when we're just doing the PK, now we can add PD. And the conclusion is that if human experimental models are applied during the phase 1, it can obtain very critical and important data, which will provide information for decisions in planning phase 2 trials. And the first answer will be the 3 now demonstrating that indeed this new chemical entity, or whether this new chemical entity has analgesic properties in humans, not only in the rodent models. And then we can also, by doing the this human experimental model testing during SAD and MAD study, we can also start to get information about the dose response properties of this new chemical entity. Another important step would be determining whether there's a predominance of peripheral versus central effects of the new chemical entity. So we are now getting to the point where we can have human data in decision making, and when we choose which model to use, we will have a better data. This also becomes very important when there are a couple of compounds that have a similar mechanism of action, or more than a couple, when a sponsor has to decide which compound to move forward. During this very effective time, during phase 1, it is possible to provide, again, human data on the basis of which to make the decisions. So I hope I have given you good insight into role of human experimental models in early therapeutic development of analgesics, and they hopefully can facilitate translation from preclinical discoveries into new clinical therapies. So I will now conclude my presentation, and Dr. T will tell you more about our capabilities and experience. Dr. T.
Thank you, Misha. My name is George Atty. I'm the Vice President and Medical Director and Site Director for the Worldwide Clinical Trials Early Phase Unit in San Antonio, Texas. Just to give you a quick overview and some understanding of our facility, we are a 300-bed facility here in San Antonio. Been in business for quite some time now. I've been with the company about 4 years. I've been doing phase 1 since 2005 and have been involved in multiple protocols that involve pain models as part of the early phase of phase 1 activities. The experience that we've had with those models is obviously something that is that are very interesting to perform and to incorporate in early phase. Here at our facility, we have 3 full-time physicians that are dedicated to doing phase 1 research. We also have on-site pharmacy with 3 full-time pharmacists. The important thing there is that our pharmacy is full service, and we can perform all types of compounding, IV sterile prep. We have a Class 10,000 clean room, and we are also, just in the past year, have started CGMP phase 1 manufacturing. And that's important as when it comes to finding different ways to deliver the drug and or the agents that we are using in the pain models. We also, as a full-service unit, have a Motara surveyor system. We run about 120 protocols a year. We have extensive experience with SAD, MAD, and proof-of-concept type studies. We currently have 4 ongoing SAD, MAD programs, which is about average for us at any one time. We also can do the TQT, DDI, BE, BA, and ADME studies, and also, especially when it comes to new chemical entities that may cross the blood-brain barrier, we can do CSF sampling for PK as well as biomarkers. So when it comes to incorporating or trying to get an early signal for these studies and incorporating experimental pain models, obviously, the important part there is design of the protocol. And one of the things that we pride ourselves on is helping with the development of the protocol, as well as the decisions on how to incorporate these pain models. the reality is that that in order for these types of protocols to be successful, the protocol needs to be written with enough adaptive language so that you can follow the path as it directs you. For those of you with experience, you understand that frequently we run into barriers or issues that were unforeseen. So when it comes to writing these types of protocols, and I've helped create protocols with that are SAD, MAD, and proof of concept all in one study, and it's a challenge, but when you think about it, it sort of lays out your program the way you expect it to. The beauty of having a single protocol is obviously it allows for overlap of your SAD and MAD portion, and or which of course shortens your timeline, and the fact that by having a single protocol, you can gain some economic efficiency there. But the key is our approach to writing these protocols, and the approach to try to facilitate the best course, and at the same time incorporate all the necessary PK and PD information that is required for your program. And in some cases, and we again are very willing to work with sponsors, but performing a pilot study to get better information about the type of method that you want to use can be very helpful. And again, with Misha's input and our experience, we could definitely entertain those types of activities. Of course, once you write the perfect protocol and you have incorporated all of your the critical factors in your protocol, it's obvious that it will only be successful if we are able to recruit for that. And given our experience, we do have a dedicated recruitment team. We have about 20,000 active subjects in our database. But one of the key factors here is appropriately selecting subjects that are willing to perform these studies. I've done multiple studies that include the pain models or other sorts of studies that require some discomfort on the subject besides the standard blood draw. And the key here is always informing subjects and working with the subjects, giving them a full information package so that they understand that what's expected of them, what they're likely to experience, the degree of discomfort they may experience, and then once the protocol starts, there's a lot of hand-holding that goes into it to get them through all of the protocol procedures. I've actually had capsaicin injected in my arm, so I know what it feels like, and I've done studies that require 4 capsaicin injections over time. So that degree of dedication by our subjects is achievable as long as we treat them right, inform them correctly, and of course pay them an appropriate stipend. With regard to special populations, we also have the opportunity to secure some of these populations, diabetic pain models. We have, unfortunately, in San Antonio, an epidemic of diabetes, as is pretty much the case in the rest of the country. And we also partner with other Phase 1 units throughout the country that can provide us with opportunities to work with specific populations. But when it comes down to it, the reality is these are complex studies, and obviously any chemical entity that's being developed for the treatment of pain. I mean, there are opportunities out there to get early information from your phase 1, incorporating the pain models in your SAD or MAD protocol, and translating that information from the phase 1 into our later phases. Again, the idea here is that we're mainly want to act as partners and provide all of our expertise and or experience with these types of programs, and then taking it from the information from the preclinical, working through the phase 1, gathering the information from those protocols, and then moving it up the chain into the phase 2. With our current experience in our company, we feel that we are in a good position to provide you for that service. So at this time, I would like to go ahead and open the floor. Obviously, you'll be submitting your questions online, but I think we have time to answer some questions, and so please feel free.
Great, thank you. So we have had a handful of questions come in. If for some reason we do not get to your question, we will reach out to you individually. So the first question we have is, do you think that PD models in healthy subjects is helpful in phase 1, or do you think that it is better to go fast to PA?
Interesting question. And the question I would translate is, what's the decision about going fast to proof of concept study? And the question is, can you jump into the proof of concept studies without information that the applying PD models in normal healthy volunteers can provide? And that is the information about the dose range. If suddenly the preclinical models have been developed for diabetes or arthritis, but unfortunately, what was frequently done is, and again, most of this is in the print, but many times the preclinical model used is traumatic nerve injury, like loose ligation or partial nerve injury, and which is one mechanism of action in having one course of disease. Suddenly, it is a neuropathic pain model and does manifest with allodynia and hyperalgesia. Then I'm always puzzled why then decision is to go with post-operative neuralgia, which is infectious type of disease model in humans. And so, or diabetic neuropathy. And then we get surprised like, oh, those studies failed. Well, maybe they failed because there's a mismatch on the basis of what information we have, and then we go and make a decision to go with something else. And again, you probably know my bias by now that indeed I would err on taking, and again, it's not taking too much time because you are incorporating PD models during this phase 1 that needs to be done anyway, which is the SAD and MAD study. And this is not in addition to, but it's actually incorporating during phase 1. So you cannot, I mean, you can go as fast as you wanted to proof of concept studies, but you wouldn't be doing it without doing it phase 1. So it's not either or. You would be just adding the PD models during your phase 1. George, I don't know if you have any other comments yourself.
No, I agree with you, Misha. That I agree with that.
Great, thank you. Another question that has come in, can I do human experimental model pain study on top of my traditional SAD-MAD PK study?
As I just said in the first question, yes, that's the idea, that these are done they're actually incorporated into the SAD and MAD studies. So the benefit of it is it really does not extend the duration of the study. Certainly does for our team adds more work, but we have experience and know how to make it so the information that's necessary to obtain for PK and during the SAD and MAD studies is obtained. But in addition, we get this significant valuable information about the properties of new chemical entity, which is whether in the dose ranges being studied, this new chemical entity is analgesic or not. And I find that incredibly valuable. So it's certainly, answer is yes, would be incorporated during the phase 1.
What is your success rate with having subjects complete a pain study?
I'll take that one, Misha. I've had a very high success rate, probably in excess of 98%. Rarely will people, once they experience it once, and again, this is probably applies more to capsaicin because that is the most intense. If they've experienced it once, then they usually will finish out. But again, it's very, very important that in order to achieve that success rate, you do a significant degree of hand-holding and or coaching at the time. Not in coaching how they're answering questions or responding to pain, but just providing them with expectations and so yes, we've had a very high success rate retaining the subjects through the entire protocol.
To expand on this, to answering this question, actually, all the subjects are that are considered for enrolling into these studies in this type of trials actually are pre-screened. So, for example, for UVB, they have to be classified according to the skin type, and then applying the UVB stimulation, which actually ends up resulting in UVB burn, is highly controlled, and it's based on an amount of energy, UVB energy, that's delivered to the skin. But again, there's very fine science of how that's done. So people who are too fair or people who are too dark would be excluded just for the purposes of not being able to control this degree of UVB. And then for intradermal capsaicin, every subject before proceeding into enrolling in a study, they're pre-screened in terms of what they rate pain on injection. So subjects who rate the pain less than 4 are not included because, they're dealing with what's called floor effect. And so if you have a drug that has a very, actually has an analgesic effect, but if you're already down at 3, well, it's really, there's not much room to go down. And also on the other hand, subjects who rate the pain 9 or higher, higher than 9, are excluded because they're overly sensitive and they probably would not want to have this repeated to start with. So that's what George was referring, that once subjects have experience and once they agree to proceed with the study, they usually stay with the study then because at that point they do know what this experience is all about. The other important aspect of it is having a very well-developed protocols and according to the psychophysics science, subjects will provide answers to what they are asked. So we have a very sophisticated instructions for the subjects to provide the most reliable and straightforward answers. The questions are very clear. And so this is an important part of subject training in terms of being good reporters for these trials. So indeed, once subjects are go through the screening period, which I think in this case the screening does require determining the appropriate subjects and subjects who are able and willing to proceed. That way, once when we get going, it's very rare that people, the subjects, decide not to participate in the study anymore. And usually, when they do get out of study, it's more for personal psychosocial reasons rather than for pain itself or pain study itself.
Great. Another question that has come in is there a better model to test opioids, UVB, IDC, or CPT?
I was hoping to make a point, and also I'll take opportunity to pretty much address that now, is that we would propose to do not one model of sensitization, but actually a battery. And that way we address and cover the possibility that new chemical entity has both mechanisms of action, such as opioids, which work peripherally and centrally. And most of the studies have demonstrated opioids do have effect on UVB as well as intradermal capsaicin, as well as cold pressor test. So this is proposed suggestion would be that if you are studying a new chemical entity that is probably working like an opioid, would be to use the battery that we just talked about. So UVB as well as intradermal capsaicin and cold pressor test. And during the study to use a controlled drug with a positive effect, such as another opioid, known opioid with known mechanism of action, such as morphine or oxycodone or something like that. And then also something that would have a negative, would be a negative control, like or with limited control, such as NSAID. So basically, this becomes crossover design where we would test the effect of new chemical entity on a battery, and then interpret the results depending also how the other agents perform, such as like morphine or oxycodone as an opioid, or NSAID as a negative control or limited control. So hopefully it gives you an idea that you can use the new chemical entity alone, but to interpret the results, probably it's the best to do the battery using the controls. And probably one would consider adding a placebo, and that way one can do most rigorous science and interpret the results in the most consistent way.
Another question. What is the typical number of patients enrolled in these HEMP studies?
Usually the number would be same as for MAD and SAD studies. So it's usually between dozens to 2 dozens of subjects. And and again, similarly, as Faculty suggested, many times this had to be adapted depending on a question and depending on other issues that might come up. Sometimes the defining factor might be the PK end of it. So, so certainly it is in a range of what's usually considered SAD and MAD study number of subjects.
Yeah,
I don't know if Dr. T would have.
Right. So usually see 8 to 10 per cohort, and then depending on what how many dose escalations you expect to require to either get to MTD or just to get to where you think the therapeutic range is going to be. You can usually get by with 8 to 10 subjects per cohort. So in a typical SAD/MAD, we'll see usually 5 cohorts of 8, and then in the MAD, pretty much the same thing. And again, depending on the type of compound that you're dosing, and the expected variability in your PK results. So it doesn't really change much from a standard SAD/MAD.
Okay, great. I think we probably have time for 2 more questions, but like I mentioned earlier, if you did ask a question that we do not get to, we will reach out to you individually. So here's another question. Can I use experimental models for registration?
Straight answer is no. These studies are used solely to answer the questions as outlined, whether the new chemical entity is analgesic or not in humans, what's the mechanism of action, peripheral, central, and is there a dose-response property. That's the information that could be obtained from the human experimental models, and these questions are really important in guiding decision-making for phase 2, but they are not used for registration.
Thank you. And the final question, how does your IRB view these types of studies?
We work closely with our IRB, and we have never had any pushback from the types of studies. Obviously, they understand what their requirements are, the reasons for the type of testing, and so we work closely with them and in informing them of our plans, and they usually have a very good understanding of what we're expecting of our subjects, and we've had very favorable interactions with them in the past. So these are the types of things that it's not unusual to see these types of requirements and protocols, and like I said, they usually give us a very favorable response.
My experience is identical, and certainly IRBs would have questions, again, raising the issues in terms of how much pain and how to deal with that. But on the other hand, when it's clearly explained that, number one, this pain is usually in the range of everyday experience. Number one. Number two, it's limited in duration. And in number three, that most of what we study is allodynia and hyperalgesia, which is when the area of sensitization is induced, we study the effects of applying the mechanical thermal stimuli, and that's something that subject can stop at any time. So when they understand the elements that go into the answer is always positive, meaning approval.
Great, thank you. Thank you, Dr. Bechakonya and Dr. Atti. At this time, we would like to thank everybody for joining our webinar today. As a reminder, all attendees will receive a link to view the recording, so be expecting an email with this information tomorrow. For more information about Worldwide Clinical Trials, we invite you to visit our website at worldwide.com. We hope you enjoyed today's webinar and have a wonderful rest of the day. Thank you.